Hello, my name is Joshua Mugabi. I welcome you to Coordination. We are going to look at learning outcome B and the learning activity A and B. Let's look at the structure of a neuron, parts and their functions. Item 1. A student falls during sports and injures his arm. He notices that he reacts more slowly when touching hot objects using that hand. Task. Explain how damage to the neuron could cause slower reaction time by referring to its structure. Injury may damage the axon or myelin sheath. The myelin sheath speeds up nerve impulse through saltatory conduction. Without it, impulses travel slowly along the axon. Damaged axon terminals may fail to transmit impulses to the next neuron. Therefore, the reaction becomes delayed. Item 2. An elderly patient develops a condition where messages from the brain to the muscle become very slow, leading to poor coordination. A doctor explains that the motor neurons are degenerating. Task. Using the structure of a motor neuron, describe how degeneration leads to poor muscle control. Motor neurons have long axons that carry impulse from the central nervous system to muscles. Degeneration leads to reduced ability of dendrites to receive signals. Slow transmission along the axon due to myelin loss. Weakening of axon terminal causing poor neurotransmitter release. As a result, muscle receive weak or slow instruction from the brain, leading to poor coordination. Item 3. Students dissect a rat's spinal cord and observe long thread-like structures extending from the spinal cord into muscles. Task. Identify the part of a motor neuron and explain how each part contributes to transmitting nerve impulses. Cell body, soma, contain nucleus, controls cell metabolism. Dendrites receive impulses from other neurons and send them to the cell body. Axon, long fibers that conducts impulses away from the cell body to effectors. Myelin sheath, insulate axons, speeds up impulse transmission. Nodes of Rabia, gaps in the myelin where impulses jump, speeding conduction. Axon terminal, synaptic knobs, release neurotransmitters into the synapse. Synapse, junction where impulses pass chemically to the next neuron or muscle. Item 4. During a lesson on reflex action, a student asks why impulses travel so fast from the skin to the spinal cord during withdrawal from something painful. Task. Using the structure of sensory neurons, explain why reflex action are rapid. Sensory neurons have long myelinated axons. Myelin sheath ensures saltatory conduction, leading to very fast impulse transmission. Dendrites detect stimuli in skin and send impulses to the cell body quickly. Cell body positioned off to the side allows direct flow of impulse along the axon. Axon terminals quickly pass impulses to the relay neuron in spinal cord. This structure design ensures rapid reflex responses. Item 5. In class, students use a nerve model to demonstrate how electric impulses travel. They note impulses only travel in one direction. Task. Using neurostructure, explain why impulses do not travel backwards. Neurons are polarized, meaning impulses travel from dendrites to the cell body, to the axon, to the terminals. Synapses allow transmission in one direction only because neurotransmitters are released only from axon terminals and receptors are found only on the next neuron's dendrites. This structure prevents backflow of impulses. Item 6. A certain poison blocks neurotransmitters releasing at the synapse. A patient exposed to it becomes paralyzed. Task. Explain, using neurostructure, how blocking neurotransmitter release causes paralysis. Neurotransmitters are released from axon terminals. If blocked, impulses cannot cross the synapse to the next neuron or muscle. Muscle fibers receive no signal, therefore do not contract. Paralysis occurs because messages from the central nervous system cannot reach effectors, which are the muscles. Item 7. Students examine diagrams of different neurons in their books. Task. Describe structure differences between a relay neuron and a motor neuron and relate them to their functions. Relay neurons. They have a shorter axon and many dendrites. They are located entirely in the central nervous system. Under function, they connect the sensory and the motor neurons. Motor neurons. They have long axon extending to muscles, fewer dendrites at cell body, axon terminals at muscles. Function: carry impulses from the central nervous system to effectors, which might be muscles. Differences: reflex roles in the reflex arc. Item 8. A patient is examined after a car accident. Doctors discovered that many dendrites in the patient's brain neurons were damaged. Task: explain how dendrite damage affects nerve communication. Dendrites receive incoming impulses. Damage reduces the ability to receive and integrate signals. This slows down or blocks communication between neurons and may lead to loss of sensation, memory issues, or poor coordination. Now let us look at impulse transmission and varying stimulation. Item 1. Student touches 3 surfaces: a smooth table, warm water, and a sharp pin. She withdraws her hand faster from the pin and warm water than from the table. Task: explain why the nervous system responds differently to these different stimuli. Stronger or harmful stimuli, sharp pain, high temperatures, trigger more frequent action potentials. Pain receptors and thermoreceptors generate impulses at a high rate. More impulses reach the central nervous system, thus faster reflex response. Weak stimuli, like smooth table, generate fewer impulses, leading to slower or no reflex. Item 2. A student walks from a bright classroom into a dim corridor. He notices that his eyes take time to adjust and impulses initially seem slow. Task: explain impulse transmission differences in strong versus weak light. In bright light, photoreceptors, the cones, are strongly stimulated, producing frequent impulses to the brain. In dim light, photoreceptors, the rods, generate fewer impulses at a slower frequency. Adjustment time is needed for the retinal cells to adapt and restore sensitivity. Item 3. During an experiment, students clap softly near a subject and gradually increase sound intensity. The subject reacts faster to louder claps. Task: explain why louder sounds result in faster response. Louder sounds leads to stronger stimulation of the hair cells in cochlea. Stronger depolarization result into higher impulse frequency. Central nervous system processes impulses faster, thus giving a quicker reaction. Now let us look at factors affecting impulse transmission speed. Item 1. A student reads that pain from burning heat is felt faster than pain from deep pressure on the skin. Activity: identify the structural features that make some nerve transmit impulses more quickly. Myelinated fibers transmit impulses faster than unmyelinated fibers. Larger diameter axon conducts impulses more quickly. Higher temperatures increases impulse speed. Shorter synaptic delays in certain pathway enhances conduction. Item 2. Two nerve muscle preparations are tested. One kept warm, the other cooled. The warm preparation shows rapid contraction, while the cold one contracts slowly. Task: explain how temperature affects impulse transmission speed. Warm temperature increases enzyme activity, leading to faster sodium-potassium pump function. Faster opening and closing of ion channels leads to quicker depolarization and repolarization. Cold temperatures slows enzyme action and ion movement, thus slow transmission. Item 3. A patient with a degenerative disease loses myelin around motor neurons. Task: explain how loss of myelin affects impulse speed. Myelin allows saltatory conduction. Impulse jumps between nodes. Without myelin, conduction becomes continuous and very slow. This results in delayed responses and weak muscle movement. Let's look at significance of warm-ups and cold packs. Item 1. A class conducts an experiment measuring reaction between before warm-ups, after warm-ups, and after applying ice. The conditions and the average reaction time in minutes, microseconds. Before warm-up, Reaction time was 260 microseconds. After warm-ups, 180 microseconds. After cold packs, 361 microseconds. Task: analyze the data and explain the effect of warm-up and cold on impulse transmission. Warm-up from 260 to 180 microseconds. Temperature increases results in faster enzyme activity. Faster opening and closing of ion channels leads to quicker action potentials. Increased blood flow improves oxygen and nutrient supply, resulting in shorter reaction time and faster response. Cold packs, that is coming from before warm-up 260 to application of cold packs 310 microseconds. Temperature decreases leads to slower sodium and potassium pump action. Synaptic transmission becomes slower. Receptors and muscles respond sluggishly, resulting in longer action time. Item 2. A netball coach insists athletes warm up for 10 minutes before playing. Some students do not understand why. Task: using nerve impulse transmission principles, explain the importance of warm-ups. Warm-ups elevate body temperature, leading to faster nerve impulse conduction. Muscles receive signal more quickly, hence enhanced coordination and reaction time. Faster synaptic transmission results in quicker decision making. Reduced injury risks because muscle respond rapidly and efficiently. Item 3. A sprinter twists her ankle and a first aid volunteer applies a cold pack immediately. Task: explain why cold reduces pain and slow impulse transmission. Cold reduces temperature around nerves, leading to slow ion channel function. Fewer and slower impulses from pain receptors reach the brain. Cold reduces inflammation and swellings, thus less pressure on sensory nerves. Results: pain signals slow down and discomfort decreases. Item 4. A hiker with a frostbite reports that she barely felt pain when the skin became extremely cold. Task: explain explain neurologically why very cold temperatures reduces sensation. Extreme cold slows or stops action potential formation. Ion channels freeze or slow drastically. Myelin becomes less efficient. Sensory neurons transmit very few impulses, resulting into reduced perception of pain. Let's now talk about advanced information. We shall look at the structures of neurons. We have different types of neuron, a motor neuron here, magnified with the different parts. Here we have the sensory, motor, and relay neuron. This is a relay neuron. We have a sensory neuron here, and then we have a motor neuron. Let's talk at the reflex arc. Reflex arc, we have a pin here on the hand, and we have impulses go through the sensory neuron, the cell body of the sensory neuron. It goes to the spinal nerve, and then it enters into the cell body of the relay neuron. Then from there it connects the cell body of the motor neuron, and it comes back through the axon of the motor neuron, and it comes to the nerve ending of the motor neuron, which makes contact with the muscle. That one is going to cause what we call a reflex arc. Patella reflex. Patella reflex, we have a hammer here and a stimulus which is acting as a stimulus. It is knocking, and when it is knocked on the patella, there are stretch receptors which will receive the information through the sensory neuron. It will go to the dorsal root ganglion, then to the relay neuron in the gray matter here, then from there very fast synapses in the spinal cord, and it comes back through the motor neuron. Then the motor neuron takes it to the muscles. Cross-section of the spinal cord and the three types of neuron. So we are looking at the cross-section. Here we are going to have our sensory neuron. Sensory neuron comes and connects synapses with the relay neuron. Then the relay neuron synapses with the motor neuron, which brings back the messages to the effector, which is the muscle. Impulse transmission at the synapse. Here we have this is the axon of the presynaptic neuron, and then here we have the presynaptic membrane. This is a postsynaptic neuron with a postsynaptic membrane. We have receptors here. We have inactivators, cholinesterase here, and we have here the vesicles, which contains the neurotransmitter, the mitochondria for provision of energy, and then we have those small ones, the neurotransmitter like acetylcholine. We have the neuron cell membranes when they are polarized at rest. Now here we can see we have the membrane. During polarization, the inside is negatively charged and the outside is positively charged. And the sodium-potassium pumps, these pump using active transport, they move 3 sodium out and then 2 potassium inside. So 3 sodium go out and then 2 potassium go inside. This one is going to make the membrane become polarized. And here we have the potassium ion channel, which is always opened, and it is allowing the escape of potassium ions to diffuse out. Here we look at the neuron cell membrane become depolarized when they are stimulated. So we have our curve here. At this point here, this is repolarization, and here there is going to be stimulation, meaning we are depolarizing. After depolarization, then we shall have the action potential here. After the action potential, there will be repolarization up to this point, and then we shall have hyperpolarization at this point, and then we shall take it back to resting potential, which is the repolarization. But this part here where we have hyperpolarization, that one is what we call refractory period. Action potential moves along the neuron. So when the stimulus arrives at this point, the inside becomes positively charged, which we call depolarization, and we have the action potential. Meanwhile, the other part of the neuron is still repolarized. The inside is negatively, or which is still having a resting potential. Now, when the impulse has passed, then outside becomes positive and inside becomes negative. That is hyperpolarization. During that period is what we are referring to as the refractory period, as the action potential is moving along the neuron. A synapse is a junction between the neuron. Here we have our synapse. This is the presynaptic neuron and the postsynaptic neuron. It has got vesicles, that is the presynaptic neuron, has vesicles which are filled with a neurotransmitter. And then the postsynaptic membrane has got receptors. And the gap in between them is what we refer to as the synaptic cleft. Okay, now let us look at biology assessment. There is what we call formative assessment, and this includes observation, whereby watching learners working, assessing the skills, values, and attitude. Then there is a part of conversation. This one you ask questions and talking to learners. Here we are assessing the knowledge and understanding. Then there is the product part. The product part appraising the learner's work. This can be in form of writing, report, translation, calculation, presentation, map, diagram, model, drawing, painting, and many others. A product is something physical and permanent that can be kept and looked at. Thank you very much for participating in coordination learning outcome B, analyzing the impulse transmission in relation to the structure of the neuron, a chemical synapse, and the factors that influence the neural activity in response to environmental stimuli. We have been looking at the learning activity A and B, where we're saying that the learners should search about the structure of the neuron to identify the relationship between the parts and their functions, and they make their own noted illustration and present them. And then they should also look at the impulse transmission factors affecting impulse transmission speed, and the significance of warm-ups before physical activity, and the use of cold baths in first aid. Thank you very much for participating. If this has been so useful to you, please visit me at Mugabi Joshua Biology on YouTube.